Continuing with the same topic, inflammation. We ended our last lecture with acute inflammation and saw the predominant cell type in acute inflammation is neutrophils. Now, if the inflammation persists for a longer period of time, say more than a week, then the inflammation has now entered into chronic inflammation stage. Now, the most cell types which are found during chronic inflammation are the macrophages. These macrophages are formed as a result of differentiation of these monocytes, and as well as there will be some lymphocytes present. Okay. Now, again, the properties, the physical and the chemical properties of the biomaterial or your implant will suggest how long the chronic inflammation will last. Okay. Yeah, it can be of short duration of time, but again, short term is relative in nature. But inflammation will be remaining confined at the implant site. Inflammation always happen locally. The presence, if the surgeon sees the inflammation is present or the chronic inflammation is present for a very long period of time, like more than few weeks, maybe months or years, then there will be a final conclusion that there is an infection growing when brewing, okay, at the like behind your implant. The role of lymphocytes, as I mentioned, lymphocytes is one of the cell types which are present in the chronic inflammation. Their role is not really known, okay, not completely elucidated in literature, but their role would be, as you can estimate, would be antibody production, and these antibodies can help or then act as mediators of immune response. Similarly, macrophages process and present the antigen to the immunocompetent cells. That is, the macrophages are the antigen presenting cells. They are going to present the antigen to these lymphocytes, and then because of that, antibody production can happen. Macrophages, they are the very, very important cells in chronic inflammation because of the large number of bioactive products it can produce. So it can produce various kind of proteases, right? Then chemotactic factors or the chemokines. Then free radical oxygens, moieties, as well as complement components like C3b, if you remember the example that we discussed. Then it can also produce various coagulation factors, then growth promoting factors, as well as cytokines. So, which is why macrophages stay for a very long period of time. Their concentration remains consistent. If you remember the graph, which shows the temporal sequence of the events happening, and these macrophages, as they're staying for a very long period of time, they continue to function by producing or synthesizing these uh, types of signaling molecules, enzymes, etc. The growth factors that are produced by these macrophages, it can in turn stimulate a wide variety of cells, which will initiate their cell migration, differentiation, and ultimately tissue remodeling. Now, let's assume that the inflammation ended with the chronic inflammation step. Now, like the body thinks that it has cleared the foreign body, right? But in reality, it doesn't really happen for the implant. It is true for small bacteria or fungi, etc. If they have entered, so they can be easily removed, and the end of the inflammation means the end of the removal from the body. But for biomaterial implant, which are much bigger in size, for them, inflammation basically continues. All right. How it continues in what now what it will be called, we will see now. Okay. And in this case, the formation of granulation tissue, how does it happen? Now let's discuss this from this slide onwards. Now, within 1 day following implantation, the healing response is initiated by the action of monocytes and the macrophages. Agreed, just like we saw in the previous slides. The fibroblasts and the endothelial cells, they are present at the implant site. Why they are present? Because they are being called, right, through various growth factors. And since they are present, they will be stimulated to proliferate and then ultimately form the granulation tissue. So if the granulation tissue is like has started to form, that means we are moving towards the healing part now. All right. The inflammation step was to contain the infectious agent. All right. A wall of the infectious agent. Now, after inflammation, the healing part will begin. And the formation of granulation tissue marks the beginning of the healing. So usually, we have seen granulation tissue actually in normal wound healing. Like if an injury happened, like a small cut, etc., we have seen granulation tissue. It looks like a pink, soft granular appearance. It has a very pinkish appearance. Okay, soft, and it's also very soft. You have to protect it. All right, you cannot prick something sharp in it, otherwise another wound will start creating. Why it is like that? It is because the fibroblasts, they will start secreting this ECM, which is relatively new. All right, it has not really cross-linked very well. All right, then also the blood vessels, they are now the cells are now forming new blood vessels, and blood flow is quite new. So that is why the pinkish appearance comes in. Depending on the extent of injury, the granulation tissue, it can like start forming as early as 3 to 5 days following implantation. Again, depending on how badly the site is injured. Okay, remember I discussed that the homeostasis, if it is like disrupted too much, then we cannot reach the formation of granulation tissue within 3 5 days. It can take months or years. The new blood vessels, which are formed from the pre-existing one, it happens through the process of angiogenesis. You already know this. We have already discussed this process in our previous discussions. This process involves proliferation, maturation, and organization of endothelial cells into blood capillaries. Now, fibroblasts, they also proliferate, and while they are proliferating, they are also synthesizing ECM components like collagen and proteoglycans. And these ECM components, along with the fibroblasts and endothelial cells, they form the granulation tissue. The protoglycans concentration can be seen very high in the early stages, whereas collagen, then in the later stages, the collagen concentration it increases. Okay, because uh, so it increases, and this collagen contributes to the formation of the fibrous capsule in the last step, that is fibrosis. Some fibroblasts, they have features of smooth muscle cells, all right, and they are like specialized fibroblasts, and they are called myofibroblasts. And these myofibroblasts, they help in wound contraction, as seen when the granulation tissue starts to develop. Again, we have discussed this. This wound contraction is mediated by the myofibroblasts when we were talking about the wound healing process, normal wound healing process. The wound healing by primary union, or the first intention, is the healing of clean surgical incisions in which the wound edges have been approximated by surgical sutures. So wound healing will happen faster if the cut that is being made by the surgeon is very clean and the edges are smooth. But if the like cut happened very abruptly with very rough edges, then the sequence of events, the time required for each event to happen, that will vary. So wound healing by secondary union, a second intention, it occurs when there is a large tissue defect that must be filled. So basically, they are trying to say uh, for clean and nice surgical wounds, the wound healing happens like fast, all right, and it is called wound healing by primary union. Okay, whereas the wounds which are created as a result of some accident, because of which our body or that particular site has lost a tissue, large amount of tissue, it is damaged. So then the wound healing will the the way the wound will heal, it is happening by a secondary union. So it is just a nomenclature to distinguish that for surgical wounds, the wound healing will happen by primary intention is just a correlation. All right. And for tissue defect, where due to accident, large amount of tissue has abruptly been defected or damaged, then the wound healing will happen by secondary union. So in the second case, due to the loss of tissue, the fibroblasts and endothelial cells, so they will have to work a lot to synthesize that much amount of ECM. And that too, when this amount of ECM gets synthesized and the cells remodel that particular area, the original architecture cannot be really reconstituted the way it was before the injury. Okay, and this kind of wound healing that is happening by secondary union in this kind, uh, the amount of granulation tissue formed is very large, and as a result, fibrosis that too happens to a very large area, and we get larger scars. Okay, now this figure that I have shown here in the slide, it shows the cells, predominant cells that is present at different locations. Okay, and how do they come to that particular location and what cellular fate processes they undergo. So in the blood, there is monocyte. So by when the injury has been created, they will migrate as a result of chemotaxis to the tissue. So in the tissue, the monocyte has arrived. Now, since your implant is maybe composed of some biomaterial, so as a tissue and biomaterial interface, these monocytes, like 10, 15 monocytes, they will fuse together and form macrophage. And how this is happening? This is happening through the process of differentiation. Now, these macrophages will also adhere to the biomaterial surface, so adhesion will take place, and subsequently try to cover up the entire area through migration and chemotaxis. Now, these macrophages are responsible for secreting various kind of signaling molecules, so they will be responsible for signal transduction in their neighboring cells, as well as if the biomaterial is very large, that it cannot be phagocytosed, then various macrophages, they will fuse together and differentiate to form the foreign body giant cell. So this is how the cells will look like at different stage. This foreign body giant cell is the largest cell with large number of nuclei. Now this foreign body giant cell, it will remain adhered to the biomaterial surface and continue to try to phagocytose it. Okay, so this is the cell type which people usually find when they remove the implant after certain duration and see these cells being adhered on it. And the foreign body giant cells adhered on the implant, it is itself, the entire system is itself encapsulated by a fibrous capsule that is formed through the process of fibrosis. That is the last step. Now, our material cannot be phagocytosed. Okay, so the macrophages, they give a frustrated phagocytosis response and creates what is called foreign body giant cells. So these foreign body giant cells themselves will try to again phagocytose the material, and if they cannot, then like they will continue their efforts. Okay, but uh, in reality they cannot. So, for example, breast prosthesis, the silicone breast prosthesis. So, when people remove it from the body, they find these foreign body giant cells around it, and they are continuing to secrete various proteases and try to degrade it. But since the material is not responsive to those enzymes, then how it will degrade, right? So, formation of the foreign body giant cells and their activity is called the foreign body reaction, and that happens in presence of large non-phagocytosable implants. So foreign body reaction is composed to biomaterials is composed of these FBGCs and the components of the granulation tissue. So the foreign body reaction uh, involves the foreign body giant cells. Obviously, they're the main players here, as well as some components of granulation tissue, like the blood vessels or the capillaries. Okay, then the fibroblast, then the macrophages, other macrophages which have not fused. They also in part contribute to this reaction. So the extent of this foreign body reaction, it depends on the form, topography of the surface of the biomaterial. All right. So if the surface is smooth, such as in breast prosthesis, then the foreign body reaction will be composed of a layer of macrophages that is one to two cells in thickness. So um, it is not that intense. That is what I'm trying to say for smooth surfaces. Whereas for rough surfaces like vascular prosthesis, there the reaction will be composed of this foreign body giant CS as well as various large number of macrophages. So this will indicate thicker cell, uh, more layers of cells, all right, and the reaction has happened more intensively. Whereas fabric surfaces, they generally respond to the presence of fabric surfaces. They are also the foreign body reaction will be composed of macrophages and the giant cells. So for smooth surfaces, only the foreign body giant cells will be present, and it will create a layer around it. Means outer layer around it, and it will be around one to two cells in thickness. That is not too thick, not too many cells are involved. Whereas for the other type of surfaces like the fabric surfaces or the rough surfaces like vascular prostheses, for them more cells are involved, which is why the cellular thickness or the outer covering of cellular thickness is greater. It consists of this foreign body reaction consists of macrophages and the giant cells, and it can persist very, very long time at the tissue and implant interface. At least it can persist for entire lifetime of the implant, for however long the implant is supposed to stay in the body. And in general, what happens after a certain amount of time, our body tries to isolate it. So how it does, it like the fibroblast cells, as I mentioned, they secrete collagen in a very large amount, and this collagen is responsible for creating a capsule, also called fibrous capsule, around the that surrounds the implant, and with it, the interfacial foreign body reaction which is going on. Thus, it isolates it from the local tissue environment. So the healthy tissue which is there around the implant that doesn't get affected by the continuous reaction, foreign body reaction happening. So it is very, very common to see these foreign body giant cells at the surface of the implant throughout the lifetime, but it is not clear whether these cells they remain active means throughout their lifetime or they become like quiescent means silent. They just remain adhered because maybe they don't know where to go, so they are like अच्छा ठीक है मैं ही सही है, maybe like that, or they like continuously working hard on the same goal to phagocytosis. So that thing is not clear to us yet. Then the last event that happens to the biomaterial, that is the fibrosis step on the fibrous encapsulation. Now here what happens, uh, the collagen capsule gets created. Now something else is written here. No, so let's read these two lines. Repair can involve two processes, either replacement of the injured tissue by the parenchymal cells of the same type, or replacement by connective tissue that constitute the fibrous capsule. So, as tissue engineers, we are mostly interested in the first process. Means we want to like deliver the parenchymal cells of that particular tissue type. Parenchymal cells basically refer to the functional cells that individually express the function of the tissue. For example, like epidermis, that epidermis layer of our skin. So keratinocytes of the epidermis layer, they are the parenchymal cells. Okay. Then similarly, cardiomyocytes, these are the parenchymal cells of our heart. So I'm talking about that. So we are mostly interested in facilitating the repair process by delivering these cells at the injured site. Or we can trigger the proliferation of these parenchymal differentiated cells so that enough number of cells come and repair the region. Or if that is not possible, second way of repairing is through formation of connective tissue, which mainly composed of fibroblast and the ECM components like collagen, elastin, etc. And if this step happens, then fibrous capsule gets constituted. These two processes are controlled by the proliferative capacity of the cells in the tissue receiving the implant and the extent of injury, or the persistence of the tissue network, that is framework, at the implant site. Means which of these two repair process will happen depends on these two factors. Okay, so cardiomyocytes they don't proliferate, right? So if you take these cells out from the heart and try to culture it, they won't grow. Similarly, hepatocytes, the human hepatocytes, they will also not grow. So in those cases, the most obvious answer how the repair will go on is through the second process. Okay. Or if you are implanting in such a way that you're disrupting that tissue architecture very much, okay, then large amount of fibrous tissue will start forming instead, and it will not be because that area is not sufficient, or the area cannot be filled up by the proliferative, like by the proliferating parenchymal cells. So it cannot create that much amount of parenchymal cells needed to fill that defect area, which is why which process will happen. Uh, it will depend on this particular factor as well, second factor. The regenerative capacity of cells it depends on their type, whether it is labile, stable, or permanent. Labile means those cells which continuously multiply and divide throughout their life. Stable means they divide only when needed, whereas permanent cells they are not able to replicate at all. Okay, so these are if we distribute cells based on whether they can replicate or not, then these three in this way we can classify those cells. So you can understand a perfect repair will happen when the labile cells are present and the stable cells because they have the capacity to undergo multiplication, that is proliferation. Whereas if the injury has happened at tissue site, which is composed mainly of the permanent cells who themselves cannot replicate, then ultimately, in order to fill that particular defect area, our body will give a green signal for fibrosis to happen. The condition of the underlying framework means the architecture of the tissue and what kind of cells does it have. That will play an important role on whether the normal architecture of the tissue after the wound repair, after the wound will be restored or not. All right. So if the framework is not properly, if it completely destroyed, if the tissue framework is completely destroyed due to an accident, then then the path that our body will take to repair it is through fibrosis. Otherwise, if the tissue network or tissue framework is somewhat retained, slightly damaged, not very much damaged, then perhaps our body will take the path of completely reconstructing. Not perfectly, okay, somewhat it will be imperfect, but it will try to reconstruct the tissue the way it was before the wound healing, but not perfectly. Okay, so that's it on this topic, that is post response to biomaterials, and these are the two references which I have used for preparing the notes on this particular topic. If you ever need it in future, you can maybe go through these references and get more knowledge out in this area. Thank you. I'll see you in the next class.